Have you heard of neurology, the study of brains? How about paleontology, the study of fossils? Did you know that you can combine these two into one discipline? If you're interested in brains and fossils, you should hear about paleoneurology. Dr. Joanna Gabrielle O'Tilly Edinger, or Tilly Edinger, was the founder of this field and the most influential paleontologist of the 20th century. But her academic success in paleontology was unusual. Not only was she a woman in academia in a time when very few women were, but she was also deaf. What's more, Dr. Edinger was Jewish, and her life and research were upended by living in Germany during the Holocaust. While the world seemed against her, she persevered, founding the field of paleoneurology and discovering the secrets of long-dead organisms. Paleontologists study the history of the Earth through fossils. Fossils are rocks that contain remnants of living organisms from long ago, and there are many ways that they could form. Fossilized remnants can be things like bones, shells, hardened footprints in the mud, or the imprints of living material that has since decayed away, such as plants or animal tissue. Paleontologists use fossils to study what types of plants, animals, bacteria, insects, and fungi lived on Earth at various stages of the Earth's history. They can also learn about how the climate, ecosystem, and geology of a region has changed over time. But there's a big problem with learning about these extinct organisms from fossils. It's very rare for soft tissues to be fossilized. Soft tissues include things like muscles, skin, and organs, which decay much faster than bones, teeth, or shells. This can make it super difficult to learn about ancient animals' bodies. Paleontologists have to come up with clever ways to get information from the fossilized clues they dig up, and Tilly was as clever as they come. Tilly was born in Germany in 1897. She began to lose her hearing as a teenager due to a disease called otosclerosis. She was fascinated by the brain from an early age since her father was a neurologist, and this passion led her to earn her PhD at the University of Frankfurt in paleontology, geology, and zoology. During many years of the Holocaust, Dr. Edinger had to carry out her research by sneaking into her work building and hiding her hearing disability. She even had her name tag removed from her office to avoid drawing attention to herself. She tried to move to America, but was initially denied because of strict immigration quotas. In 1940, she was approved to bypass the quotas and to move to America because she was hired as a research associate at Harvard University. She was only one of 4 female scientists who were granted this kind of aid. Dr. Edinger became a U.S. citizen in 1945 and came to think of Boston as her home. As a paleontologist, Tilly was fascinated by the soft tissues of ancient organisms, the kind of tissue that isn't fossilized and is lost to time. But Dr. Edinger realized that although the brain decays, it can leave an imprint on the skull that can tell us how its surface was shaped. She came up with the idea of using these fossilized skulls as molds. By covering the inside of a skull with rubber or plaster, you get a replica model of the organism's brain. These internal casts of a hollow object are called endocasts. Tilly had made a huge discovery. Endocasts could be used to reconstruct parts of the neural anatomy of ancient animals, allowing paleontologists to study their brains, almost like they would study any other brain. Tilly used this method to learn about a dinosaur called the Nothosaurus, which was a marine reptile from the Mesozoic era. The Mesozoic era was between 66 million and 252 million years ago, so the information that their fossils hold can be very important to learn what life was like for creatures in the Triassic, Jurassic, and Cretaceous periods. As part of her research, Tilly studied a natural brain cast made of fossilized mud that had been discovered inside a skull. This inspired her to start making endocast molds and found the entire field of paleoneurology. While Dr. Edinger worked at the Harvard Museum of Comparative Zoology, she researched the evolution of another organism, horses. By studying the endocasts of their brains across a single geological time era, she tracked the subtle changes in size and surface folds that progressed over thousands of years. Since evolution happens over such long periods of time, this kind of study would be impossible to conduct on preserved brain samples, simply because brain samples haven't been collected or preserved for a long enough time. Dr. Edinger's use of fossil samples and endocasts uncovered entirely new knowledge about neurology and the evolution of mammals. Brains are really complex with lots of internal structure, but we know different regions of the brain process different things. For example, our sense of smell comes from a section called the olfactory lobes, and for whales, this region sits on the outer layer of the brain. Tilly studied the skulls of whales throughout history and found that the indents made by the olfactory lobes got smaller over time, indicating that their sense of smell was reduced. This evolution makes a lot of sense, since whales are mammals and it's pretty hard to smell underwater when you breathe air. Dr. Tilly Edinger founded an entirely new field. Paleoneurology was born from her unique passions in multiple subjects. Throughout her career, she followed those passions, found new questions to explore, and mentored many students in this budding new field. She even has a bone named after her, the Tilly bone. Dr. Edinger thought creatively and found a way to do what seemed impossible: study the brains of long-gone animals. Countless secrets of the ancient world were unlocked through Tilly's dedication to bones and the stories they tell.